to now what we are calling innate immunity. So as we've seen, we've looked at the anatomy of the immune system in general. We've seen how it is and where the immune system is mostly distributed, either in the primary lymphoid organs or the secondary lymphoid organs. We've also seen what we are calling the barriers, or rather natural factors that are present that prevent any infection. So let us now move to the innate immunity. The innate immunity. And so the innate immune responses, their main activity, or their main role, of course, is to respond, or being the first line of response, as some of you are calling it in the previous class, to foreign invasion. But as we were discussing, we saw that, okay, fine, as much as we are focusing on foreign invasion, the immune system does not necessarily focus on foreign invasion. It also has to find a way of focusing on itself, because the body is also producing its own self-antigens, which have to be dealt with. They have to be recognized by the immune system. If they are not recognized by the immune system, then you can't differentiate self from non-self. And so such scenarios would maybe result into autoimmune diseases. We've seen that. But mainly, so we're looking at the foreign invasion, but also saw that we have other non-infectious diseases, which of course the body of course deals with them. And it deals with them, especially if you're talking about cancers. Cancerous cells are going to express unique antigens on their cell surface. So in such a case, that unique antigen will be seen as a foreign agent, and it should be dealt with. In most cases, it is a bit difficult, because then the immune system is unable to differentiate whether this is just a normal antigen on the cell or it's a foreign antigen, and it's bypassed, of course. And even these infectious agents, the parasites, the bacteria, part of the reason why they are problematic is that they are able to mimic the way the body behaves, and of course they escape all immune response tactics, including of course developing resistance to most of these treatments that are present currently. So, but the body would have an innate immune response to first of all deal or be a first line of defense. And clearly these are present at birth, innators. Somebody called them congenital, but we are referring them to be present at birth. So of course, they are active during both the initial and subsequent encounters with the foreign substance. And characteristically, they are primitive and stereotyped. So we are saying primitive because they have not specialized to focus on a specific antigen or on a specific invader. They are still primitive and stereotyped. So they deal with a big array of, say, bacteria, a big array of fungi, a big array of whatever it is. Then, of course, to differentiate it with the adaptive immune response, there is no memory. There's no memory. We know that in the adaptive immune response, antibodies can be made and memory can be retained for the infectious agent, so that if there is a secondary invasion, then there's mounting of an immune response very quickly. But that is not present in the innate immune response. So, so the innate immune response, or the innate immune system, recognizes structures on foreign substances that are known as PAMPs. It recognizes PAMPs, and PAMPs are called pathogen-associated molecular patterns. I think we talked about it previously. I think this is the part I was ending the class with. And we said, so we have PAMPs that are on foreign invaders. And these PAMPs are recognized by pattern recognition receptors. So these PRRs, or pattern recognition receptors, are present on innate immune cells. So the innate immune cells have PRRs that recognize foreign invaders, and these foreign invaders have pathogen-associated molecular patterns, or PAMPs. Okay, so these PRRs are present on cells, but PAMPs are present on the foreign invader. Maybe it's a parasite, maybe it's a virus, maybe it's a fungi, maybe it's a prion. That prion would have a PAMP. Now, the cell that is innate, the cell that is an innate, will have a PRR, and PRR are going to recognize this. Once they recognize these PAMPs, then an immune response, of course, is triggered to prevent the invader. So these PRRs are present on, say, macrophages. They are present, of course, on neutrophils. They are present on what we call natural killer cells. And those three cells are the cells that we call cells of the innate immune system. So they are white blood cells, yes, but would classify, or rather categorize them as cells of the innate immune response. Now, or innate immunity, or innate immune system. We have an idea that once an infection occurs, the first cells at the site of infection are the neutrophils. So they are likely part of the innate immune response. We also seeing the macrophages, and I've also talked about a type of cell, white blood cell, we are calling natural killer cells. So NKs, or natural killer, we'll talk about them a bit in a few, probably today, before we just end the class. So they are key. But the most important thing is that these cells, what you are calling cells of the innate immune system, have pattern recognition receptor. And the pathogen, or the invader, has the pump. So PRRs will detect pumps and then trigger an immune response for preventing that invader from getting into the cell. Most of the times it's successful, but sometimes, of course, it fails. But of course, many agents are usually prevented from entering cells, from entering the body at this particular level of the innate immunity. So because of that, most of the immunology, or the immunology study, is so little on innate immunity and so much on adaptive immunity. So adaptive immunity, of course, is more organized, more specific, less primitive, less stereotyped. So we can understand the mechanisms there better rather than the innate immune system. So in innate, usually we just brush off, we look at it briefly, then go to the adaptive, but it's an important area. For instance, I've talked about the MALT. MALT is currently a very serious area of interest, and people are really discovering many things that we previously did not know every other time in the MALT. So just wanted to display this schematic here, not to scare you, but to make you understand that some of the things that are going on the cells. So this I picked it from an article in Nature, and so this is just a cell. This is the cell membrane. If you're able to see, this is the lipid bilayer, of course, of a cell membrane. So this is an innate cell. Innate cell, it could be a neutrophil, it could be a macrophage, it could be a natural killer cell, but not necessarily natural killer, because natural killer cells are a bit specific. So, and again, I was insisting in the previous class that we say that this is non-specific, but I'm cautious on how I'm using that term, not really insisting on it being non-specific, because we are going to see that they are also specific to some extent. Much as it's stereotyped, it is a bit specific to some extent. Now, I'll jump. Let's see if. No, let me just introduce this, then I'll elaborate this in details in a moment. So, for instance, we're able to see that this is TLR, TLR, TLR. I talked about TLRs last time in class. Can somebody remind us what TLR stands for? If you're taking note in the previous class, I talked about TLR. TLR.
Toll-like receptors.
Toll-like receptors. Toll-like receptors, correct. So this TLR has toll-like receptors. Now, these toll-like receptors are these pattern recognition receptors. These are just pattern PRRs. So if you talk about a TLR, it is a PRR. It's a pattern recognition receptor. Now, this is a neutrophil. And in this neutrophil, it has toll-like receptors of different types. We have 1, we have 2, 3 iko wapi? Yeah, 3 here, 4, 5, 6, 6 iko wapi? 6, 6, 6. Nani amo? 6 is here together with the 2. 7 is here. 8 is here together with 7. 9, 10 iko wapi? Kuna mtu ameona 10? 10? Hakuna 10. There is 11 here. Okay. So these receptors are about 10. 10 to 11 thereabout. Has anybody seen 10 from this structure? Has anybody seen toll-like receptor 10? Anyone? Oma, why are you crying? Has anybody seen toll-like receptor 10? In a moment, you're going to just, they're not complicated things. You'll understand what they do. Yeah, so don't look at the structure and get intimidated. Just simple structures, you'll be able to understand what they mean in a moment. But the key thing here is that we know that toll-like receptors are PRRs. They are simply for pattern recognition receptors. And the work of pattern recognition receptors is to detect pumps. They are able to detect pathogen-associated molecular patterns. That is what they are supposed to do. Now, so what I want to elaborate here is this. For instance, toll-like receptor 4. For toll-like receptor 4. It's a CD14. Toll-like receptor 4, CD14 ni neutrophil. So toll-like receptor 4 recognizes a pump called lipopolysaccharide. Okay? Toll-like receptor 5 recognizes a pump called flagellin. Toll-like receptor 11 recognizes a pump called profilin. Toll-like receptor 9 recognizes DNA, but this DNA has to be CPG. This DNA that is CPG is full of cytosine and guanine. Toll-like receptor 3 recognizes a pump called double-stranded RNA. This must be a virus. This toll-like receptor 7 and 8 recognizes single-stranded RNA. Toll-like receptors 2 recognizes what we call a peptidoglycan. So this is recognizing bacteria. This is recognizing bacteria. So toll-like receptor 2, this thing is called a peptidoglycan. This thing is called a lipopolysaccharide. These are all present on cells of bacteria, and they help us differentiate between what we call gram-positive and gram-negative bacteria. Gram-positive bacteria, such as tuberculosis, has this thing I'm calling the peptidoglycan. on the cell surface. So when you stain it with gram, it becomes blue in color. So it's called gram-positive. Lipopolysaccharides, instead, they do not take up the stain. So bacteria that have lipopolysaccharides are referred to as gram-negative. So this is able to detect what we call gram-negative bacteria. This is able to detect gram-positive bacteria. Now, if you have bacteria that have flagella, we know flagella that is present on especially prokaryotic cells, then tool like receptor 5 is able to detect that this is flagella. Human beings don't have flagella, then this is an invader. Okay. If you have a virus that is double-stranded RNA, in human beings, RNA is single-stranded. So double-stranded RNA will be detected as foreign, and that is the molecular pattern that is going to be picked. Human beings do not have DNA that is full of cytosine and guanine. It is balanced. So if you see DNA that is full of CPG DNA, then that is foreign DNA. The body is able to detect it as foreign and so on and so forth. So of course, there's a cascade. We'll briefly talk about this cascade because immunologically, we have to understand the mechanism. But this just to give you a broader picture of what these PRRs are. So there are 10, rather 11 in number currently. I'm not aware of any new discovery that has been made. George, unaweza mute ama uko na swali? George William. Yeah, so that is in simple terms what this structure means. Of course, it will enable you to further dig deep and understand what happens. So we've said innate immune system, it is characterized by its specific cells here. These specific cells have PRRs, and these PRRs are able to recognize what? Pumps. They're able to recognize pumps. Now, if the innate immune system has PRRs, if the innate immune system has its cells, I talked about neutrophils, I talked about macrophages, then it means the innate immune system has a mechanism for which it works. It has a mechanism for how it works. So, of course, eventually I'll share with you these notes. You'll go to this article, mepeana link hapa. You read in details what PRRs are, what they do, and it will enable this article. Of course, this is where I got this, but there's more information about this. So you're able to read this information at this particular page. You'll be able to, it's a free article, of course, in Nature. So Nature is, of course, one of the top journals in the world, scientific journals. So if you go there, you'll be able to read this and you'll be able to understand what these PRRs do, what do they recognize, and so on and so forth. So one thing about, as you're doing your microbiology, one thing you'll know that these invaders, these infectious agents have unique things about them, have unique markers about them. For instance, most viruses have RNA instead of DNA. For instance, HIV is RNA, single RNA. Corona is single RNA. But you have viruses such as hepatitis, hepatitis B is DNA, hepatitis C is RNA. So based on that, there are very good PRRs that the body can be able to detect. So besides that, the innate immune system will use 3 major mechanisms to prevent foreign invasion. The first mechanism being phagocytosis. I'm sure you've talked about phagocytosis before, especially in your physiology. Inflammation. I'm sure you know what inflammation is. We didn't even define it in the previous class because we already have encountered it. And as you are doing pathology, as you're understanding pathology, you're also going to do inflammation in details. So yes, it's cross-cutting both in immunology as well as pathology and pathology and immunology are interrelated, of course. You can't understand diseases without understanding the mechanisms for which those diseases present. And in most cases, some of these diseases are mechanistically, you can be able to understand them either from a biochemical angle or from an immunological angle. You're able to then understand how these diseases develop and how they cause the body to respond. So these are interrelated. So I'll not dwell so much on some of these things, such as inflammation, for instance, because you're covering it elsewhere. But we know that inflammation is response to injury. Then cytotoxicity. Cytotoxicity, of course, is in elimination of infected or transformed cells via apoptosis. So apoptosis also helps a key in elimination of this. And apoptosis, of course, is programmed cell death, as you mostly know. So we talked about cells of the innate immunity. And most of these cells are phagocytic cells. So they are phagocytic cells. It means they have PRRs. They have this. Basically, this cell of the innate immune system is phagocytic, yes, but it will also have this. So the phagocytic cells or phagocytes that are involved in the innate immune system, the first one being neutrophils, and neutrophils will commonly refer to them as polymorphonucleus leukocytes or PMNs. So neutrophils are neutrophils because of this. We are able to tell them because of how this lobed nucleus looks like. We know that they are the most abundant white blood cells. They are multi-lobed, even. See, the lobing is just quite a lot. And they are usually the first cells at the site of infection. So they are the most abundant white blood cells. They do not divide. They are short-lived. They are multi-lobed. And they contain granules. Now, these granules are important because these granules then help us to differentiate. So there are two main types, and these granules are able to, again, enable the cell to kill or rather interfere with the invader and ultimately kill it. So we have two types of granules. So ideally, this is how the neutrophil looks like, as a multi-lobed nucleus. That is how what makes it different. And then within the cytosol or cytoplasm, we have granules, and we are saying we have two types of granules. So these two types of granules are the primary azurophil granules, and the primary and remember, we're talking about cells that are multi-lobed. So this is how the nucleus looks like. So within the nucleus, we just have these two types of granules, the primary and the secondary. Primary, I think, are blue in color in this schematic. Secondary are yellow in color. And we also have a tertiary granule that is not very prominent. So the key granules are these. So the primary one is called the primary azurophil granule. And this primary azurophil granule develops quite early, and it contains myeloperoxidase. So it contains the enzyme myeloperoxidase, meaning it is able to oxidize membranes of bacteria, for instance. So it has an oxidative way of killing using radicals, but it also contains non-oxidative antimicrobial effectors such as defensins. You remember we talked about defensins. Tulisema defensins was in a deal na nini. Defensins, if somebody can remind us what defensins do. Defensins, if you remember. We saw that defensins are able to deal with pressures.
Bacteria, fungi, and and enveloped viruses.
Correct. So, so defenses are quite broad. They're able to deal with all that. Bactericidal permeability increasing protein (BPI). It is present in azurophil granules. So, what this bactericidal permeability increasing protein does is that, of course, it then creates holes within the cell wall of the bacteria, and the bacteria is able to get a lot of liquid into it, and it lyses. Then we have cathepsin G. Cathepsin is also you remember we talked about proteases, and we said proteases are proteins that cleave. There are enzymes that cleave proteins, and then it depends with the amino acid that is common in that protein. So we have what we call cathepsins. Cathepsins are proteases. We have different types of cathepsins. Kuna cathepsin A, G, K, blah, blah. There's quite a number. So cathepsin G is a serine protease. Cathepsin G is a serine protease. Now, if I say it's a serine protease, what do I mean? That cathepsin G is a serine protease. If I say it's a serine protease, what do I mean? From the explanation I've just made, of course, it's an amino acid. From your biochemistry, the serine is an amino acid, and proteins are made of amino acids. So you can't say that a protein is serine is a protein; it's an amino acid. So when we say it's a serine protease, what we mean is that it cleaves serine within a protein. So when you have a chain of protein, of course, a chain of protein is made of amino acids linked together by a peptide bond. So what is happening here is that the moment cathepsin G sees serine in the structure of a protein, it cleaves it, cleaves it. And so serine, especially, you'll find that some proteins are rich in specific proteins. For instance, you remember we talked about histidine-rich proteins, ABA, that in saliva, histidine-rich proteins. So it means this protein has other amino acids, but there's a lot of histidine in it. And these are unique proteins for especially fungi. Most fungi have proteins, yes, but these proteins are rich in histidine. That is the unique thing. Now you'll find that some bacteria are present, but these bacteria are rich in, say, a specific amino acid. It could be arginine, it could be serine, like in this case, it could be, yeah, any other. So what happens is that in this case, when this bacteria sees any serine, it cleaves, meaning it then prefers to cleave bacteria that are rich in serine, and by their proteins are rich in serine. So by that extension, it will kill it. So for instance, now, yeah, cathepsin G is a serine protease, and this serine protease would cleave bacteria cells, cell walls, sorry. We also have, yeah, hata pia cathepsin A, I think it's a serine protease. Cathepsin K is a protease also, but I don't know, can't remember what. I think it's, yeah, aspartate. It's an aspartate. It focuses on aspartate, and many other, cathepsin H, cathepsin J, cathepsin L. So we have different types of cathepsins, but just like I've talked about PRRs, a cathepsin is just a protease. A protease is an enzyme that cleaves proteins. Peroxidase. Remember, the azurophil has myeloperoxidase. So what do they do? contain? Then they contain lactoferrin. We already see lactoferrin inafanya. We looked at that previously. Lactoferrin. So okay, for us to move quickly, let me skip that. But of course, lactoferrin is the iron, so preventing bacterial enzymes. These are enzymes that create, rather lead to lysis from the word. Alkaline phosphatase. Alkaline phosphatase. This enzyme, of course, will remove phosphate groups. Will remove phosphate groups from phosphate, of course, is the one that is a form of energy. Na kama energy inatolewa, then of course, if it's a process that is going on, it will not happen. So it's starving the cell of phosphate. And we know that phosphate is being emanated from ATP, and ATP will be the source of a kind of energy. Then, of course, they're also bound to cytochrome B588. I do not want to go into that particular point. So we have these components, and these components are antibacterial by nature. They are antibacterial. So when these granules are released, then, of course, they're also going to cause damage to the invaders. The other phagocytic cell, of course, is the macrophages. The macrophages are usually mature monocytes. Of course, they originate from monocytes. Now, monocytes are blood-based. Macrophages usually are not blood-based. They are tissue-based. So it starts from, I think maybe you talked about it in hematology. I didn't teach you hematology, so I don't know whatever you covered. All I did is set an exam, but I believe you covered it adequately based on how you performed. So monocytes are where we start from. And these monocytes ultimately can move to tissues. Once they move to tissues, they mature and they are referred to as macrophages. And macrophages are they are the true phagocytes. Macrophages are the true phagocytes. Just like I said, dendritic cells are the true APCs. The cell that is made to phagocytose anything is a macrophage. So the moment a macrophage moves to the tissue, it then changes its name. It changes its name. We no longer refer to it, okay, it's a macrophage, yes, but usually the name changes. Now, for instance, monocytes have moved to the lungs and they become part of the lungs. They have become, of course, macrophages, and we refer to them as alveolar macrophages. Okay, that is in the lungs. In the liver, we refer to them as Kupffer cells. In the spleen and lymph nodes, we are calling them line sinusoids. We refer to them as mesangial cells when they are in the kidney. The brain, tunaita microglia. Of course, bone, we call them osteoclasts. So I'm sure you've encountered these terminologies, or rather these types of cells before. But so if you're talking about the liver cells, so you're not just talking about hepatocytes per se. If the hepatocyte, it has the ability to phagocytose, then it's called a Kupffer cell, and so on and so forth. So the brain, we call them microglia. In the bone, osteoclast. In the kidney, mesangial cells. So they are given names depending on the tissue they are occupying. So unlike neutrophils, we saw that neutrophils are short-lived. Remember, they are non-dividing and they are short-lived. That is what we said about neutrophils. Macrophages are long-lived and they possess significant rough surface ERs and the mitochondria. So they do have this, the endoplasmic reticulum as mitochondria also, because they are large cells and they the true phagocytic cells. So I know the process of phagocytosis. Maybe we'll discuss it in this class, but I'm sure you've also encountered it before. Then, so the main function of neutrophils is to prevent or rather defense against bacteria, but the kind of bacteria we refer to as pyogenic, or bacteria that forms pus. The main role of the macrophages is they are effective against bacteria, viruses, and protozoa that are intracellular. So this will phagocytose the cell that is infected. It is most likely going to phagocytose the infected cells because they mainly deal with intracellular pathogens. So maybe neutrophils are good at dealing with extracellular because they phagocytose smaller invaders. Macrophages will phagocytose large cells that have invaders in them. Okay, that is what usually happens. But again, we'll find protozoa are large enough, macrophages are likely either to phagocytose. If they don't phagocytose, they usually form what we call a granuloma around it. Yeah, they form a granuloma around it, especially in terms of worms and tuberculosis. Macrophages are likely to form a third of the world's population has TB. Of course, we have TB in our latent form, but that TB or those cells, bacteria, are present, but they are held together by a granuloma formed by macrophages. So the moment the immunity wanes, or rather goes down, then the granulomas are usually released and an individual is likely to suffer from tuberculosis. That's why one of the key characteristics of TB is most prevalent in people who are immunocompromised. We'll talk about immunocompromisation later on in this course. I think we talked about immune deficiency. Now I want us to go back to the PRRs in a bit of detail because they are key signals in terms of phagocytosis. So we looked at phagocytic cells, and the main phagocytic cells here we're talking about neutrophils and macrophages. So these phagocytic cells or cells of the innate going to trigger a reaction that definitely eventually end up being phagocytic. But these pumps are usually typically polysaccharides or polynucleotides, and they usually differ between the pathogens. So you'll find that one pathogen might have maybe a polysaccharide. For instance, I've talked about bacteria that would have either peptidoglycan or a lipopolysaccharide. You'll talk about polynucleotides, you find that maybe viruses are going to have nucleic acids, what we are referring to as polynucleotides. So the differences are there, and that's how then these cells are able to differentiate between the pathogens, and these pumps, of course, are not present in the host. So we do not have these pumps. So therefore, the innate immune cells are able to differentiate self from non-self. So, and again, these PRRs are lectin-like. The PRRs are lectin-like. Lectin, of course, is a if you remember biochemistry, lectin is a carbohydrate. So some cells have lectin on their cell surface, but human beings, our plasma membranes, do not have lectin. So since we don't have lectin, then we're able to differentiate ours from pathogens, from invaders. So what happens is that these receptors, of course, the work of receptors is to bind ligands. Ligands are these pumps. So they're able to bind them. And once they are bound, once PRRs are bound to pumps, then there is a cascade that is initiated. There's a signal transduction that is initiated through NFKB, nuclear factor kappa B, nuclear factor kappa beta. This is a transcription factor. And with this, through this nuclear factor kappa B, then that is a signal to initiate phagocytosis. I know as nurses, you don't really go into what we call signal transduction at your physiological, when you're studying your physiology, because again, physiology is wide, but maybe you touch it a bit. But what you know that is that cells interact with each other, cells interact with foreign agents, cells interact with cytokines, cells interact with chemical signals, and so on and so forth. So the interaction triggers the action, of course. So we have what we call secondary messengers in the body. We have secondary messengers in the body. Now, let me just return you back here. Let me return you back here. We have secondary messengers. For instance, my D88. Another one is cyclic AMP, cyclic adenosine monophosphate. These are usually called secondary. No, this is called an adapter. And this adapter, this MyD88, is supposed to link to a secondary messenger. Supposed to link to a secondary messenger. Now, secondary messengers will include things like the nuclear factor kappa B, it's a transcription factor, something like cyclic AMP, it's a secondary messenger, and so on. There are very many chemicals that are called secondary. So we are not doing a signal transduction class, but I just wanted you to appreciate that, you see, this toll-like receptor, for instance, is bound to an adapter, and this adapter is supposed to be linked to a secondary messenger. And now, through a specific secondary messenger, a specific message is passed on what to happen. So what we are simply saying here is that for phagocytosis to occur, the signal has to go through nuclear factor kappa B. If the signal doesn't go through nuclear factor kappa B, then phagocytosis will not occur. For instance, if this my D88 was to link to cyclic AMP, what it means is that this cell, if this cell is a neutrophil, it's going to attract another neutrophil to the site. That is what will happen. But if this MyD88 links with now nuclear factor kappa B, then it means this cell will perform a function of phagocytosis. I hope that point is clear. So I try to simplify it as much as possible. So PRRs have to link with an adapter through this nuclear factor kappa B, and phagocytosis is triggered. So we said toll-like receptors are PRRs. Toll-like receptors are PRRs. So for you to be able to understand this, you'll also be linking with the schematic that I have, plus the link that I gave you that you go and read on your own. So it's a family of at least 10 transmembrane proteins. And we counted, we saw that we didn't spot number 10. Ama kuna mtu aliona number 10 kwa hizi? Is there anybody who saw number 10? We just saw that number 10, I told like receptor 10, I didn't see it. So they're about 10. So maybe 10 is not there. Ama you can search and see what 10 does. So either there are 10 or 11 thereabout. And probably maybe a new one has been discovered. I don't know. I've not gone through that. So of course they recognize pumps. Now, for instance, toll-like receptor 2, and they are very specific again. So we said innate immunity is non-specific. But again, here we are saying they are very specific. So toll-like receptor 2, for instance, recognizes gram-positive bacterial peptidoglycan. This. Okay. Then toll-like receptor 4 is for gram-negative bacteria. It recognizes lipopolysaccharides. And gram-negative bacteria usually produce what we call endotoxins. So endotoxins are toxins within the cell. Most gram-positive bacteria usually produce exotoxins. They produce toxins outside the cell. Then we have toll-like receptor 3 and toll-like receptor 5 recognize double-stranded RNA. So those are the pumps they recognize. So once they recognize, they trigger a biochemical cascade. That biochemical cascade activates nuclear factor Kb. And once the nuclear factor Kb has been activated, it leads to the synthesis of pro-inflammatory cytokines, as well as the peptides we talked about. And then, of course, this will be the link between innate and adaptive. The moment cytokines have been generated, then the adaptive immune system is been alerted that, well, there is a foreign invader here. Probably you're not going to deal with it completely. Adaptive immune system, can you come into play? So this is exactly what is happening here. This is what is happening. This is CD14. CD14 is a neutrophil. Tulisema white blood cells zote ni CD what? White blood cells zote ni CD? Who can remind us? White blood cells are CD what? Victor?
CD45.
CD45. Yeah, then we said T cells ni CD what? T cells? T cells, pressures. Pressures we've answered. Ni Lloyd ama ni nani? Lloyd? Lloyd? CD3, yes. Then, of course, we saw CD8 and CD4. Then we saw that B cells are CD19 sana, but they can also be CD21. They can be CD22, depending on their maturity and interactions with antibodies. But now I'm saying CD14. Oh, no. So CD14 is a marker for phagocytosis. So CD14 is not necessarily a neutrophil. It can also be a macrophage. So CD14 is the unique marker for phagocytosis, actually. So phagocytic cells, that is macrophages, any phagocytic cell would carry CD14 on its cell surface. So this is leptin binding protein. Leptin, you remember leptins are unique. They are present on cell walls. LPS, of course, stands for lipopolysaccharide. Now, from my explanation, lipopolysaccharide, is it now a gram-positive bacteria or a gram-negative bacteria? From the explanation I've just had. I know you're going to deal with this, especially when you're doing microbiology in details, but now, is it a gram-positive, is it a gram-negative? Anyone? Tumesema nini just a few minutes ago? Valerie? Valerie? Valerie, you didn't raise your hand? Valerie, Lloyd, you can answer it so that we save a bit of time. Lloyd? Sorry? Gram negative. Valerie, unasema?
It's gram negative.
Okay, so we just said, yeah, this is a gram negative, meaning it is recognized via toll-like receptor 4. So this is the cascade that is triggered. So this is a gram negative bacteria because it has a lipopolysaccharide. Now it's going to interact with toll-like receptor. This toll-like receptor will be toll-like receptor 4. This toll-like receptor 4 will interact with a kinase, a serine 3-on kinase called IRAC. IRAC will interact with MyD88, which is an adapter. This process of adapter interaction with toll-like receptor and IRAC, of course, this is a kinase. It means a phosphate has been added here. It will trigger another molecule called TRAF. This is a tumor nucleotic factor, receptor associated factor. Then TRAF will trigger NEEC. NEEC will trigger IKK. IKK will be cleaved into IKB, and IKB is bound to NK nuclear factor KB. Once this happens, then nuclear factor KB is activated, then they separate. And once they separate with IKB, because this is now phosphorylated, this undergoes ubiquitination, it undergoes degradation, and nuclear factor is triggered, and the transcription factor now triggers the expression of pro-inflammatory genes. So pro-inflammation is triggered here, but again, phagocytosis is initiated. So key thing here is that I do not want you and cram this. This is just for you to appreciate that. So as we are talking about nuclear factor KB, nuclear factor KB is not just activated immediately. It has to be phosphorylated. And in its phosphorylation, it doesn't get phosphorylated directly. There has to be other factors, other proteins that are involved, including IRAC, TRAF, NEEC, IKK, and IKK is then cleaved to become IKB. IKB is bound to this, then it's activated. Once it's activated, IKB breaks off, phosphorylated, and is degraded. Then this is this, which toll-like receptor is activated. And then, of course, how nuclear factor KB comes in. Mnaweza ruka hii story yote and see how these pro-inflammatory genes are activated. But just for you to be able to appreciate that it's not just a simple thing as activation of nuclear factor KB, it's a cascade. So we talked about a biochemical cascade, and this is the cascade you're talking about. All these nuclear factors and the transcription factors are involved so that then the nuclear factor KB is activated. Now, so once that happens, then of course the phagocytosis occurs. Yes. So remember here, there is expression of pro-inflammatory genes. So pro-inflammatory genes are going to express so that then inflammation can occur. We know what inflammation is. Remember the three mechanisms we talked about in terms of innate immunity. So of course, inflammation is one of them. So pro-inflammatory genes are triggered to now initiate inflammation. But in addition, there is phagocytosis that is occurring. And in phagocytosis, if you're dealing, say, with a cell, we know that there's the initiation stage. I'll not go into the details of phagocytosis because I'm sure you've also covered it previously. So then, so there's this initiation. Of course, there's contractile system that is activated, actin and myosin. There is a pseudopod formation. The pseudopod is then formed. Then it's going to, of course, engulf, and ingest. And within the pseudoform, of course, there's usually a phagosome, and that phagosome would contain partly the granules that we talked about. Remember, the azurophil, the primary and the secondary are the primary azurophil, and then the secondary one is of course, it's non-peroxidase granule, and they all, of course, would release their contents as they kill, but the cell, of course, has been ingested phagocytically. So that is how then the killing happens. So there's phagocytic killing, but there's also radical killing, and radical killing we're going to discuss in the next class, of course. So using, of course, ROIs are going to also perform the killing. Yeah, so I think that is enough for you for today. Maybe I would welcome a few questions, a few clarifications before we call it a day. Any questions, any clarifications that you'd want? So let me assume that we are good. And if you are not good, you know what to do.